All right, class, welcome to another demonstration recording. Today, I am demonstrating how to graph your pigment extract data. Okay, so remember, what do we have going on in this lab section? You've taken a 96 well plate. You've loaded up a well with your isolated pigments. You should have loaded up 4 wells, okay? One for carotene, one for chlorophyll A, one for chlorophyll B, and one for xanthophyll. Your data is going to come out in this format, all right, with a bunch of wavelengths running on the left column, okay? This will cover the entire spectrum, okay, of visible light. And then you're going to have an absorbance value for each of those wavelengths. So what the 96 well plate reader has done is it has measured in each well at every 5 nanometers absorbance. It's a very fast process with the 96 well plate reader. You used to have to do it step by step in a spectrophotometer. That'd be a lot more work. Okay? Imagine blanking every time you adjust the wavelength of light. So anyways, you may need to label these columns with the appropriate pigments, but you should know which well has which pigment in it. Make sure that you've labeled each column with each pigment. All right? Cool. So I've already done the heavy lifting in this Excel sheet right here, where I've broken up into each bench, okay, the wavelength values, and then the different pigments. It's easiest if you put the wavelengths of light to the left of your pigment data, your absorbance data, like I have done right here. Okay, great. This is an easy one. If you've done the heavy lifting and the hard work of setting your data up into columns like so, then all you should have to do is click the upper left, okay, the wavelength title, scroll down, hold shift, and click the lower right cell, okay, making sure that you have all of your data encapsulated. Then you can go to insert at the top of the screen, and then right here you'll see the chart. I'm sorry, the cursor capture doesn't work here, so you'll have to just look at the top here. Insert X, Y, or bubble chart, a scatter plot, and I like the scatter plot that has the smoothed lines. Okay, here you go, scatter with smooth lines. Now, our hard part of this lab is going to be cleaning up this figure, because it has everything we need, but it's kind of a mess. So let's go ahead and edit it a little bit. First thing, click the grid lines, okay, and hit delete. Get rid of those grid lines. Next, that x-axis is horrible. It has all sorts of, it has a crazy range of empty values. Now, we only looked at wavelengths from 420 to 750 nanometers. Your lab may have looked at different wavelengths, and that's okay. You'll just need to change your x-axis. So click the x-axis, and then right-click the x-axis. And now you will have an option to format the axis. Click that button, and that's going to open up this window that's floating above my face right now. Okay? You can change the minimum bound to, in this case, 420 nanometers, and the maximum bound for us goes up to 750, so just adjust that as well to 750 nanometers. Hit enter, and now the data is actually, the figure doesn't have a bunch of empty space. You don't need a chart title here, okay? Click that chart title and delete it. Get it out of here. You do need axis titles though, right? So go ahead and click the little green box to add chart elements, or you can click this box in the top left to add chart elements, as long as you are in the chart design portion of chart tools. So we're going to go ahead and add in our axis titles. For our y-axis, we're looking at absorbance. Okay. Our x-axis, all right, is looking at wavelength. And our wavelength is in nanometers. Okay. Now it's all looking pretty good. if you set it up with the labels in the data, you even have an effective legend here telling you what each wavelength is. And if you remember from lecture and from the lab, this is pretty similar. This set of data, which was from a class in the past that I taught, this is a good set of data. You'll see we got peaks of absorbance over here. I can't highlight it, but peaks to the left side and peaks on the right side. And then that region where there's very little absorbance, that's going to be what correlates to the green portion of the color spectrum, the visible spectrum of light. So that's why these plants look green, because they don't absorb green light. Now, let's go ahead and get into putting this into a nice little PDF format. In my lab sections, I'm requiring a PDF upload. So just follow along with me here. Select the figure. Control-C to copy it. Open up a new Word file. Control-V to paste it. There it is. It's looking great. Select the figure, right-click, and insert a caption. This is figure 1. I like to throw a period after figure 1, and then call it something like absorbance spectra for primary plant pigments. Okay. Data collected. This will be what? Let's say October 19th of 2023 at Cal Poly Humboldt. Just to get in the habit of typing out decent figure captions. Throw a period at the end of that thing. Hit OK. Oops, yeah, hit OK. And now you've got a nice figure caption underneath the figure. Okay, here it is. It's looking pretty good. Now I have View Ruler selected. So you hit View at the top and make sure you check the little ruler box in case your data doesn't look the same. Because then what you can do is you can resize the figure to whatever size you want. And still, if you click within the caption and then click the ruler up at the top, you can adjust the bounds of the caption. You want to make sure that however you size your figure, the caption doesn't extend beyond it, just to make it look nice. Okay, cool. That's it. Then go to File up at the top left, save it as an Adobe PDF. Yeah. Great. I'm just going to save this as my desktop. As demo. Okay. And then save it as demo. It'll save it as a PDF. You'll be good to go. And then your PDF's going to look like this. That's a nice PDF. That's a good submission for my money right there. Okay, it's just that easy. We kept this video pretty short, I think. Ask your TAs questions if you need assistance. I hope this was helpful, and I hope you have a great rest of your day. All right, goodbye.